This is part one of a two-part lecture on cell membranes. We're going to focus mostly on chapter 6, but I am going to incorporate a little bit from chapter 5. So the first part is from chapter 5, and this is just to explain why cells are so small. Why do we see larger organisms made up of lots and lots of little cells rather than one large cell? And it has to do with the surface area to volume ratio. So this lecture is on cell membranes and how things move in and out of the cell, and so that's why I incorporated this in with this lecture. So the volume of the cell is where all of the chemical activity is going on. The surface area is where things can move in or out of the cell. So the surface area is really the cell membrane, and so as a cell gets larger, it needs to have bring more things into the cell, and it's going to be producing more things that are going to be exported out of a cell. And the problem comes in this surface area to volume ratio. So this slide illustrates this very well. If we look at a cell that's 1 micrometer in diameter, so right here we have a sphere that's 1 micrometer in diameter representing a cell, and we were to calculate the surface area to volume ratio, we would find that it has a 6:1 ratio. Now, if we double the size of that cell, we end up with a 3:1 surface area to volume ratio. If we triple the diameter of that cell, we end up with a 2:1 ratio. So the volume increases at a faster rate than the surface area, and that's what really makes it so that cells have to be very small. Okay, so we're going to focus on the membrane of the cell, and this is just a picture of the model of the cell and what we kind of think it looks like. And so if you're looking at this picture, on the outside of the cell there's a liquidy environment, and on the inside of the cell there's also a liquid environment, and this just shows all different components of the cell. So we have the phospholipids, which we'll talk about, proteins that are embedded in the membrane or just adhering to the surface of the membrane. This is cholesterol molecules that are embedded in the membrane here. These are different carbohydrates, and we'll talk about each of those components of the cell membrane. So we'll start with the phospholipid bilayer, and what the cells do is it kind of forms a sandwich with these phospholipids. So because the outside is a watery environment, the inside is a watery environment, the phospholipids oriented themselves in such a way that the hydrophilic heads are on the outside or on the inside with those tails sandwiched in the middle. So this hydrophobic environment doesn't involve the water. So the water's on the inside, the water's on the outside, and then we have this hydrophobic layer. And this is a phospholipid, which should look familiar to you from our biochemistry unit. Okay, the membranes are fluid, they're in constant motion, and this has to do with the composition of the different lipids. So the lipids within the membrane vary. They can vary in the length of the fatty acids. They can vary in the degree of saturation, which we've talked about earlier, and then also the phosphate groups. The other thing that will affect the fluidity of the membrane is the cholesterol, and this is related to temperature, and so cholesterol will help to maintain fluidity in a membrane at lower temperatures. And again, these membranes are dynamic, they're constantly changing, and we've looked at this a little bit before, but here we have the nucleus, here's the nuclear envelope, and then the endoplasmic reticulum, which is a continuation of that. And so a vesicle can bud off of the endoplasmic reticulum, fuse with the Golgi apparatus, eventually bud off from the Golgi, and then fuse with the cell membrane. So you can see membrane can be moved from place to place. So don't think of it as just a static environment. Okay, the proteins that are embedded in the membrane or that are associated with the membrane can be embedded, or they may just be associated with the fatty acid heads, the phospholipid heads. So if they do not have hydrophobic groups, then they will not span the membrane. They would just be attached to the surface of the cell membrane. Other proteins have hydrophilic regions and hydrophobic regions. And so what they end up orientating themselves in such a way that the hydrophilic regions are found inside or outside of the cell, and those hydrophobic regions are sandwiched in that hydrophobic layer in the membrane. Okay, there are also carbohydrates that can be attached to the surface, and these carbohydrates act for cell recognition from one cell to another cell, or for cells to adhere to other cells. And there are two different kinds of these. There are glycolipids, and so a glycolipid, glyco meaning sugar, is a carbohydrate that's attached to the lipid, attached to one of the phospholipids. But some of them are actually attached to those proteins that are embedded in the membrane, and we call those glycoproteins. But again, these are involved in the cell-to-cell recognition and adhesion cells adhering to other cells. Okay, so this leads us into the focus of kind of this lecture, which is in transport. So cell membranes, because of all these different components that we were just talking about, are selectively permeable, meaning that they can allow some things to pass through, other things not allowed to pass through. Sometimes some things can pass through some of the time, but not all of the time. And we divide these types of transport into passive transport and active transport. So in active transport, they do not require energy, and that's going to be the focus of the rest of this lecture. The next lecture will be on active transport. Okay, the first type of passive transport, I think most students have a handle on. It's pretty intuitive, and you've experienced it, and you're familiar with it, and that's just diffusion. So molecules move from where there are more of them to where there's less of them. And it kind of goes along with our entropy from the last unit. Things are going to go to randomness or to disorder. So here we have all of these inks that have been put into this beaker, and so we have the red all in one area, the green all in one area, and the blue all in one area. And if we let this sit, over time, molecules are going to move from where there are more of them to where there are less of them until you have a random distribution of the molecules. So, and again, it's not like movement's going to stop, but there's not going to be any net directional movement at that point. Okay, the rate, or how fast diffusion occurs, depends on a couple of things. One is the diameter of the molecules, or the ions. So the larger diameter, the slower the diffusion. The temperature also affects diffusion rate. We know temperature affects the kinetic energy of molecules, so if they're moving faster, diffusion is going to speed up. If we lower the temperature, diffusion rate is going to decrease. And then finally, concentration gradient. So if there's a higher concentration gradient, diffusion is going to occur at a faster rate. As you reach or getting closer towards equilibrium, the rate would then decrease. Okay, the one concept here that students have a hard time with is osmosis. Now, in its simplest form, osmosis is just the diffusion of water. So water is going to be moving from where there's more water to where there's less water. The confusion thing is that it's counterintuitive, because we don't usually define the concentration of something by the solvent. We define it by the solute. And so now we're talking about the movement of the solvent, which makes it a little bit tricky. So let's look at a couple of scenarios. So here we have a scenario where the cell is placed in a solution that has more dissolved stuff in it than the cell does. So we're going to call this a hypertonic. Hyper means high, tonicity is concentration, so a high concentration. And these are relative terms I'm going to be introducing to you. So we have to say what's hypertonic and what's not hypertonic. So it's going to be hypertonic relative to something else. So in this case, we're saying that the outside of the cell is hypertonic relative to the inside of the cell. So there's more dissolved stuff outside of the cell than inside of the cell. So water will move from inside of the cell to the outside of the cell. So we have to kind of look at this and say, okay, there's more dissolved stuff here. That means there's less water, relatively speaking. Less dissolved stuff here, more water. Water's going to go from where there's more to where there's less. I think the easiest way to get your head around this is to just think water follows solute. Water follows sugar, however you want to think of it. So the water is going to go to where there's more dissolved stuff. And if you think of it that way, you won't get tripped up. So if I think of it that way, I just say, okay, there's not as much dissolved stuff in the inside the cell than outside of the cell. Water is going to leave the cell. Now, how plants and animals deal with these different tonicities we're going to talk about varies. In a hypertonic solution, plant and animal cells do not function very well. So they do what's called plasmolysis. So as We take this animal cell, a sugar, red blood cell in this case, and say put it in a very high sugar environment, water's going to leave the cell and the cell's going to shrivel up. In a plant cell, because it has a cell wall, it doesn't really shrivel, but the cell membrane will pull away from the cell wall, and ultimately that will lead to cell death in both of those situations. Okay, the next type of environment would just be an equal concentration across space, and so we call that an isotonic environment. And in this case, animal cells are very happy. This is where animal cells like to be, in an isotonic environment. But plant cells do not function as well. So plant cells would actually go limp or flaccid, or kind of think of wilting in that kind of a scenario. But here we have an equal distribution across space. And then the last type of tonicity is a hypotonic solution. Again, relatively speaking, we're going to say that the outside of the cell is hypotonic relative to the cell. And so that means that there's hypo means below, a lower concentration outside of the cell than inside of the cell. Remember, water follows sugar, water follows solute, so water's going to go from where there's less of this dissolved stuff to where there's more. So water follows solute, water's going to enter the cell. Now, animal cells, this is not a good thing. So animal cells don't have a cell wall, so as water enters and enters the cell, eventually the cells will burst, and that's called cytolysis. Plant cells have a cell membrane, so they actually like a hypotonic, being in a hypotonic solution. This gives them turgidity or turgor pressure, and so the water enters, and now what happens is that cell membrane starts to push on the cell wall, and that gives the plant structure. Okay, so moving away from water for a second, although we're going to bring it right back again, another type of passive transport we call facilitated diffusion. So again, this is diffusion. It's still the movement of molecules from where there's more to where there's less. So it's a passive transport. But in this case, we're talking about something that's not going to move freely through those phospholipids. So something that may have a charge associated with it, or act like it has a charge associated with it. So this could be an ion, or it could be a polar molecule. And so these will tend to move through the proteins that are embedded in the membrane. So there are two different types. There are channel proteins. And so in this case, we just have a polypeptide chain, and on the inside it'll have polar amino acids, and that acts as a pore for these either polar or charged molecules. Another type are carrier proteins, and these actually interact with the molecule that is moving through them, or that they're transporting across the membrane. So again, these are just proteins that are embedded in the membrane, it's just how they function is different. One specific type of channel that we'll be talking about quite a bit are ion channels. And ion channels are specific to the ions that they allow to transport. So there'll be a sodium channel, and that sodium will not allow, say, potassium to pass through, or the potassium channel won't allow sodium to pass through. So they're specific to the ion that they transport. And a lot of these are gated, and we're going to see this as we continue through the course, so don't get too stuck on this now. But some of these will be closed until something happens to cause them to open. And so this can be a couple of ways that can happen. One is a molecule might have to bind to the channel. So here's my little molecule binds to a binding site on that protein, causing that protein to change shape, and now allowing these particular ions to pass through. Another way is through an electrical charge changing, and these are called voltage gated. And so we get a flood of a certain kind of ion, and that may cause these gates to open or close. And we'll see that when we study the nervous system. Okay, and I'm going to end by just talking about water again. So water, again, is a polar molecule. It's small. It can move through the phospholipid layer, but not at a very high rate. And so other ways that we see water moving through the membrane is kind of by hitchhiking. So some of these ions will form these hydration shells, and so the water molecules surround that. And then as the ion goes down its concentration gradient through a channel, the water kind of hitchhikes a ride and comes through. And then the last is there are special channels in the membrane, these special water channels, and we call these aquaporins. And that's the end of this lecture. The next lecture will be on active transport or moving against the concentration gradient. I hope this helped.